Okay, thank you very much, folks. So what I'm going to talk about is actually work that was done by a postdoctoral researcher who worked with me for a couple of years, a chap called Marcus Mellin, who is now returned to his native Finland. And in addition to Marcus, the bird data that I'm going to talk about was acquired by two ex-colleagues of mine, Paul Bellamy and Shelley Hinsley, and my job is to just come here, have a great time, and scoop all the glory. So what do we know? We know that bird diversity has long been used as a surrogate for ecosystem diversity. We also know that bird diversity is affected both by the three-dimensional structure of habitat and also by the floristic composition of habitat, and that is especially the case for woodlands. And guess what? We can acquire structure and composition of woodlands through the gift of remote sensing. And there's some references. So my aim is to look at structural information from airborne LiDAR and some narrowband indices from airborne hyperspectral data to see how they relate with avian diversity. And my aim specifically are to compare the capabilities of these two very different kinds of data, get a sense of which works best, and also then to see if I can get an insight as to what is it that's actually driving bird diversity in the woodland context. So does it relate more to vegetation structure, to tree species composition, or to measures of condition, canopy condition? So my field site is in Cambridgeshire, in eastern England. This is very much an agricultural landscape, so fairly large fields and very small isolated patches of woodland in which we've done bird survey. And when I say small, my 4 woodland sites vary between 5 hectares and 10 hectares. So these are very small sites in which it's possible pretty much to map every single bird in the woodland. The woods are reasonably similar in as much as they're all primarily oak and ash woodland with some field maple and elm. And they do have quite a well-established understorey dominated by hazel, blackthorn and hawthorn. So for field data collection, was carried out in spring of 2014. Spring is the breeding season in the UK for woodland birds, or for all birds in fact. So basically a structured walk takes place through each of the woodlands, covering the entire woodland in which all birds encountered, that's either seen or heard, are marked on a map. That process stops if weather conditions are not appropriate, i.e. strong wind or heavy rain. It follows the common bird census process, which is common across quite a lot of Western Europe and North America. And in theory, the systematic walks that were followed on all 4 occasions should encounter breeding territories of all birds across the woodland. So that data was marked on a map and then eventually digitised and therefore made digital. So in total, across these 4 woods, we actually have records of roughly 1,400 sightings of birds across 27 different species, and I've given just indicated some of the more common birds that we encountered. It has to be said these aren't as spectacular as the toucans that I was watching at breakfast this morning from the breakfast area, which is incredible. But anyway, that's what British birds look like. So the airborne remotely sensing data were acquired from the same platform simultaneously and at the same time as the bird survey was taking place, so in June 2014. Therefore, default conditions. The LiDAR data were acquired with an ALS50 sensor, importantly 2 pulses per metre squared, up to 4 returns per pulse. And processing was pretty standard to derive a terrain normalised point cloud. For hyperspectral data, so they were acquired simultaneously at the same time as the LiDAR. The sensor used was the Aisa Fenix or Fenix from Specim. 620 contiguous bands covering the whole of the visible, near infrared, and into the shortwave infrared part of the spectrum. The bandwidth gets broader as you work your way across the spectrum to maintain signal to noise ratio. Swath width is just less than a kilometre wide, and spatial resolution is 2 metre pixels. And again, fairly standard data processing here, radiometric calibration, geometric correction and reprojection to British National Grid, because that's the form of the LiDAR data we're in, and also atmospheric correction, so essentially normalising the spectral information as much as possible. So what we did, we took the 4 woodlands and we divided them up into roughly 30 by 30 metre cells. So that formed kind of a concentric pattern, right, working from the edge of the woodland in towards the centre. And across the 4 woodlands, there were 333 of these cells, from which we extracted 3 different bird metrics, 15 different LiDAR metrics, and I said there 17 hyperspectral metrics, but that actually should be 17 times 3, for reasons that I'll tell you in a minute. So the bird metrics were related to richness. So species richness is just a straight count per cell of how many different bird species were encountered. And then more complicated ones like Shannon's and Simpsons Diversity Index, which tells you something subtly different. For the LiDAR, the metrics we derived again per cell was the percentage cover of vegetation. So we defined vegetation as greater than half a metre, so the proportion of returns that were above half a metre above the ground. We also then derived proportion cover of shrub and proportion cover of canopy. Rather than try and impose our own height thresholds as to where shrub stopped and canopy started, we actually derived them using different height thresholds. So for shrubs up to 2 metres, 4, 5, 6 and 8 metres, and for canopy that was at 4 metres, 5, 6, 8 and 15 metres. Obviously for the modelling work we never allowed multiple of those same variables to appear in the same model, right? So you couldn't have a model that had P canopy 4 and P canopy 8, for example, because they're just variants of the same thing. But the idea was to try and let the data tell us where the shrub layer was and where the canopy was. Also, standard things, maximum height, average height, standard deviation of height, and foliage height diversity, which is a measure of vertical profile. So for the hyperspectral data, as you can imagine, with 620 spectral bands, there is a myriad of different narrowband indices that we could have generated. So we tried to focus on things that were likely to be more relevant, effectively for birds. So I've listed the ones there. Essentially, they tell you something about leaf foliar chemistry, leaf green leaf biomass, potentially stress, productivity, etc. So some of these, if you think about it, and we derive the minimum, the maximum, and the mean. So that's why I said it should be 17 variables times 3, because we have the mean, maximum, minimum per cell. And if you think about it, some of these things that relate to, say, the amount of green leaf biomass, canopy productivity, they're kind of a positive thing from a bird's perspective, right? So you would expect the maximum values of those to come out in the models. Other things relate to stress or evidence of leaf senescence. That's a negative thing, really, from a bird's point of view. So maybe you'd expect the minimum to come out in the models rather than the maximum. And I flag that now because that is exactly what happens. And then the 2 metrics at the bottom is just reflected to 2 specific parts of the spectrum, 830 and 980 nanometers. And the reason for that is for a very similar but bigger woodland in the same area, I did some tree species mapping, and they were the 2 parts of the spectrum that had the greatest amount of separability between the different species. So although we haven't mapped species directly, we've at least got something which is indicates different species, or at least variation in species. So they were the hyperspectral metrics that we used. So I've kind of said all that already, that LiDAR is telling you something about quantity of vegetation and its vertical distribution throughout the height profile. Hyperspectral is telling you something about forest canopy function, condition, things like water stress, productivity, leaf foliar chemistry, green leaf biomass, and then potentially something about species information. And this is all pointing in the direction of things that matter to birds, such as the availability of shelter, and most importantly of all, food. And bear in mind, the breeding season is the time when adult birds are largely employed in finding food and getting it back to the nest to feed their brood. So that's really what's being covered by this data set. So what we did, what Marcus did, was best subset regression, And firstly, we did it separately for LiDAR-only models and for hyperspectral-only models, and using firstly only one predictor variable, then models with 2 predictor variables, 3 predictor variables, and so on, and models with 6 predictor variables. And we selected as the best model the ones that created the lowest AIC score, and we only allowed the lowest AIC score and the least number of input variables. So we only accepted a model with more variables as being better if it lowered the AIC by at least a score of 2. Then what we did is we looked at the variables that were appearing most commonly in the LiDAR-only models and the hyperspectral-only models, and we pulled those to make combination models, so models that had some LiDAR and some hyperspectral values. And suffice to say, as mentioned already, we did not allow collinear or non-significant variables to appear in any of the models. And that's how we decided it. So results, I don't expect you to be able to read all of that. But importantly, for the LiDAR-only models, for the 3 different bird metrics that we looked at, so Simpsons, Shannon's diversity, and species richness, the models were actually remarkably consistent. So for LiDAR-only, the 2 variables that came out as important was proportional cover of shrubs, and shrubs was defined as having a height of up to 8 meters, and foliage height diversity. And again, for the hyperspectral models, again, for the 3 different bird diversity measures, the outcome was exactly the same. So the 2 best variables were Vogelmann's 2 is maximum, and reflectance at 830 nanometers as maximum. I'll explain what all that means in a minute, don't worry. For the combination models, actually each of the 3 bird metrics generated different best fit models, so there's a bit more variation here. Perhaps the best thing to focus on is that the Simpsons Index, the best model had 4 LiDAR metrics. For the Shannon, the best model had 2 LiDAR and 2 hyperspectral metrics. And for the species number, the best model had 2 LiDAR and 3 hyperspectral metrics. And the variables that were going into those models were subtly different. So by combining the data, actually different things start to appear from the data. So what does all that mean? I hear your question. So from a LiDAR point of view, what it really points to is that the more structurally complex the woodland is, the higher your bird diversity will be. And that comes out in having a proportion cover of shrubs, and quite tall shrubs as well. And foliage height diversity is showing you that it isn't just about having vegetation in the shrub layer, it needs to be an understory, right? So you need a well-developed tall shrub layer which is sitting under canopy. And by having that full vertical structure, it's essentially just allowing you more niche space, right? There are more places in the woods where birds can take shelter, form nests, and look for food. And in terms of the hyperspectral data, it shows that both condition and composition matter. They're affecting where the birds are found. So I've said I'd explain these. Vogelmann 2, Vogelmann's red edge index, it is sensitive to effects of leaf area, water content, and chlorophyll concentration. And it's the maximum value that came out. So in other words, where there's lots of leaves which are healthy, they're photosynthesizing, they've got lots of water, that's good condition canopy. The birds aren't eating the leaves, the birds are eating the critters that themselves are eating and living in those leaves. Reflectance at 830 is telling you something about species type or species variation. And the other metric that came out is the anthocyanin reflectance index, and that tells you something about leaf stress and early onset senescence. And that was a negative, it was the minimum value that came out of that, whereas Vogelmann's was the maximum. So the implications of all this, the best models were models that contain both the LiDAR and the hyperspectral data. And interestingly, there's been a few talks now at this conference that have done this. They've compared multi or hyperspectral data and LiDAR, and they've all concluded roughly the same thing, all in different contexts, but they've actually all shown that the best models are the ones that combine them both. But in my case, the combination models were better than the LiDAR models, which themselves were better than the hyperspectral models. The key thing is, not by much, right? So you're better if you've got hyperspectral and LiDAR data together, but it doesn't make that much of a difference. The improvements were only marginal. So you've got these two data types that are telling you something quite different about the canopy, and are explaining different aspects about why you expect to find more bird species in different parts of the wood, and yet actually you could just use one or the other and get roughly the same kind of models. But as this is a LiDAR conference, I will put my neck out and say if I had to choose between LiDAR and hyperspectral, I would choose LiDAR every time. And I'll explain why in a minute. The two caveats attached to this, or 2 points that explain things. Woodland structure, so I keep talking about woodland structure and composition. LiDAR gives you structure, hyperspectral gives you composition, as if they're two completely different and separate things, but they're not. Because woodland structure will be affected by the tree species composition, and in addition, factors such as the age of those trees and any disturbance regime that's happening. And don't forget, condition is much more temporally dynamic than structure. So if your woodland is mature and it doesn't have a particularly strong disturbance regime, you can use LiDAR data that's much older and you won't have much of an impact, right? But with hyperspectral data, it really needs to be from the same time as your field data, because condition will change rapidly. So that's where I would end. This is already published, but as I'm generous, I know, hopefully I know the question that Nicholas wants to ask, so I'll address it before you get there. Those of you familiar with the theory of island biogeography, I hope, are thinking, surely your patches are absolutely tiny, and according to the theory of island biogeography, it should be patch size that is absolutely driving species diversity, such that it's the smallest patches that should have the lowest biodiversity. So we tested that. So we looked only at the LiDAR data, we had the same LiDAR variables, and we added in kind of landscape ecological variables. So area, distance to edge, aspect, isolation, and effectively the results are basically the same. So patch area did not come out in any of the models, but there is an edge effect, a very clear edge effect. But the reason why there's an edge effect is because at the edges of woodland, you've got more shrubs, because more light's going in, so it's more shrubbiness. The smaller the wood, actually, the more higher the proportion of the wood that is edge, not core. So there is an area effect in that way, but it's simply manifesting itself by more understory. So the outcome is actually the same, but it's understory that is helping to drive diversity. And that's published in a separate paper as well. So at that point, I really will stop and take questions.